Hello, Cell and Gene Meeting on the Mediterranean participants. I'm Adrienne, Farid, Chief Development Officer at Century Therapeutics, and I'm very thankful to the organizers for the opportunity to be able to speak to you today about Century. At Century Therapeutics, we are designing and developing off-the-shelf iPSC-based cell therapies for hematologic and solid tumors. We have an end-to-end iPSC platform incorporating allo-evasion, iNK cell, and IT cell products. We're really in the process of designing cell therapy products that have best-in-class profiles, resisting host rejection, providing exposure to patients via redosing that we believe will lead to durable responses. The products will be available off the shelf, frozen, at any clinic and hospital in the future. We'll discuss a little bit today the leadership team and our manufacturing capability that we're building. And as I will describe, the product engine enables a pipeline of CAR T and CAR NK products leading to clinical initiation next year in 2022. So a little bit of history about Century. We were initiated in 2018. Since 2018, we've really been in a company building stage, acquiring intellectual property and key technologies for the platform, implementing and optimizing the iNK and IT platform and core technologies, and recruiting a team with deep cell therapy and oncology experience. We're up to about 100 employees at about 3 sites: Philadelphia, Pennsylvania, Seattle, Washington, and Hamilton, Ontario, Canada. This year, we're switching gears or pivoting toward preclinical development focus, compiling IND-enabling packages for the first products, including iNK cell product that I will tell you about today. And the IT cell platform has been established, and we're moving programs there toward the clinical direction as well. We're also getting up and running a wholly owned GMP manufacturing plant. In the next few years, we intend to become a fully integrated company with multiple clinical programs, a product engine, and put ourselves in a strong financial position. The leadership team has depth of experience in immunology, oncology, cell therapies, and biotech startup, and we believe this positions us for success. I wanted to mention also we have a collaboration, a partnership with Fujifilm Cellular Dynamics. This partnership provides Century with a strong technical foundation. Building on the pioneering work of James Thompson in iPSC reprogramming, we have access and use of intellectual property to generate immune effector cells via differentiation using unique, scalable, feeder-free methods. And these can be NK cells, T cells, macrophages, or dendritic cells. In addition, we have privileged access to FCDI, GMP manufacturing, and dedicated capacity for Century's products at FCDI. Just considering evolution of targeted cell therapies in cancer, we're standing on the shoulders of the very impactful transformational autologous cell therapies. These have been extraordinary in lymphoma and multiple myeloma setting. However, there are some hurdles and challenges with autologous therapies. Moving to healthy donor-derived allogeneic therapies, this has actually opened up some opportunities. However, there's still a finite ability to replicate and edit these cells, and they may end up terminally exhausted. In addition, one has to go back to the donor for healthy donor-derived cell therapies. With respect to the iPSC or induced pluripotent stem cell platform, Fate Therapeutics has been quite effective in demonstrating the promise of the unlimited replication that we have with iPSCs. And Century's iPSC-derived cell products build on Fate's work as well, engineered for allo-evasion. And so, which I will describe further. The engineering for allo-evasion, we believe, will produce hypoimmunogenic cells, avoiding rejection by the host immune system, suitable for repeat dosing, and enhancing cell persistence and durability. And we're working on the 2 platforms first, allo-evasion engineered CAR iNK cells, and alloevasion engineered CAR iT cells, or so-called true T cells. Century has decided to prioritize some core capabilities that we're building, and those are actually listed on this slide. IPSC Generation differentiation, starting with access to clinical grade iPSC lines that can be differentiated into T and NK cells. Those are prioritized for us. Protein engineering capabilities, gene editing capabilities, and manufacturing capabilities. And I'll go into a little bit more depth on all of those. With respect to gene editing, our editing process is actually shown here. We're editing at the iPSC stage via CRISPR-mediated homology-directed repair using MAD7 nuclease. These edits can be conducted sequentially in several steps, and that as the iPSCs have unlimited replicative capacity. Once the iPSC is engineered, as you see in the middle of the slide, we clone and isolate single cells, which can then be formed into a master cell bank. And this master cell bank actually can be made available for the lifetime of the product. So this is a powerful piece of the platform. And then the cells can be differentiated using different procedures into IT cells or INK cells. For CAR engineering, we're focused on single domain VHH antibodies, which allow easier creation of dual or multi-specific CARs, or including greater than one binding site for the same target. We're also investigating universal CAR approach and optimizing signaling domains to improve and enhance cellular responses. In addition, we're building a core capability in manufacturing. Our in-house manufacturing complements dedicated capacity at FCDI, as mentioned. We believe it's critical to enable fast product iteration and to have the capacity and optionality to support a pipeline. So building a plan to be operational later this year. And we're recruiting a technical operations team at this time. This complements the technical development expertise that we're building as well, a capability that we're building in process research and development, analytical development, scale up, and quality assurance. Switching gears, I'd like to show you profiles of the progressing INK products. To explain that, Century's INK cells are highly similar to peripheral blood NK cells. We're showing you the phenotypic profile here. On the right, Century's iPSC-derived NK cells compared with human primary NK cells on the left. Importantly, alloevasion editing is expected to enhance durability of clinical responses. So the left-hand side of the slide shows alloevasion edits that we are conducting. This includes knockout of HLA1, HLA2, and knock-in of an allo inhibitory receptor designed to limit host immune system T and NK cell recognition. As illustrated by the figure on the top right, knockout of beta-2 microglobulin reducing MHC class 1 shows significant reduction of CD8 T cell activation. On the bottom right in the solid line, we've illustrated PK and persistence profiles typically seen for current cell therapies, where the initial dose shows robust expansion, but subsequent doses often do not expand or have reduced expansion. The alloevasion engineering should allow for redosing with similar exposure and persistence for a second, third, or multiple doses, as in the blue dotted line. And this is really, we believe, critical to our platform. Century 101 is our first product moving that's advancing. It's a monospecific CD19 iPSC-derived NK cell product. With alloevasion engineering, we've demonstrated cytotoxicity versus lymphoma tumor cell lines, and I have 3 panels here. At left, the INK cells, not CAR modified, show K562 killing through innate receptor engagement comparable to PBMC NK cells. In the center panel, After CD19 CAR transfection, CD19 INKs kill comparable to CAR Ts. And on the right-hand panel, we've demonstrated the ability of the CAR INKs to retain activity versus lymphoma cells over multiple rounds of killing, so-called serial killing. And we've shown you 7 rounds of serial killing in this plot, but more recent data using single clones shows up to about 17 cycles of serial killing for the CD19 INK. Switching over to the T cell, product, our approach to the CAR IT platform is to utilize a so-called true T cell, that is, including expression of a trusted TCR, a TCR that doesn't induce GVHD and supports T cell differentiation, for example, one that recognizes a viral antigen or doesn't recognize peptides. So progress on the iPSC-derived alpha, beta, and gamma delta CAR true T cells is shown here. On the left, iPSC-derived IT cells kill lymphoma cells comparable to peripheral blood CAR Ts. And on the right-hand side, our current feeder-free differentiation methods produce CAR IT cells that proliferate robustly upon activation. I wanted to illustrate a little bit more about one of the features of the platform, and this is a common progenitor approach. After IPS screening and selection, sequential gene editing, and single cell cloning and isolation, we produce a common what we're calling a common progenitor cell, which will include all of the particular features that we've edited: alloevasion, safety switch, homeostatic cytokines, pet reporter, other features that we have included. And importantly, this common progenitor can be banked and can then undergo just a last step of CAR insertion to target particular monospecific or bispecific antigens that correlate with the tumor. So in this way, we really have generated kind of a product engine where we can bank at the common progenitor stage and just have a more simple last step to produce the CAR-targeted product. Our discovery and preclinical pipeline connects the biology of NK and T cell therapies, validated targets, and features to overcome the specific challenges of each disease. So beyond the monospecific CD19 Century 101 product that I've described, we are focused on prioritizing Century 102, planned as a dual targeted CD19 plus IT product to improve response where antigen loss is a factor in lymphoma. And we've prioritized as well Century 103, which is planned as a dual or multi-specific CD133 targeted INK or IT cell therapy for glioblastoma. Importantly, we'll assess administration of the cell therapy locally into the CSF or tumor compartment in GBM, where we expect that will help with limiting systemic toxicity and improving targeting to the tumor. Discovery programs target additional heme and solid tumor programs that are at earlier stage are also listed, and we're pursuing those as well. So in summary, we've made significant progress on multiplex gene-edited, alloevasive, INK and IT products at Century, supporting clinical initiation next year, which we're very much looking forward to. With the product engine enabling a pipeline of CAR T and CAR NK products tailored to address treatment needs in both hematologic and solid tumors. Many thanks on behalf of my colleagues at Century Therapeutics for your attention.